Okay, so hello everyone, and first of all, thank you for the organizing team to let me present this work. So as we all know, protein function is mediated by different interactions, such as protein-protein interactions, protein-nucleic interactions, but the one interaction that we are actually interested in this work is protein-compound interactions, primarily protein-metabolite interactions, and those interactions are driven by binding through a binding cavity on the protein surfaces, which can be characterized by different physical properties, such as hydrophobicity or aromaticity or others. And with the recent release of the AlphaFold database, we have now available protein structures at scale, which actually allows it to also characterize the proteome, the pocketome at scale and identify the diversity of the pocketome as the entirety of all pockets within a species. So we were interested in what drives the pocketome diversity and complexity in evolution, mainly through how does the pocketome differ between different species. So do we have any kingdom specificities between bacteria, fungi, plantae, and animalia? How does this scale with the number of proteins or folds found within one species? And can we link that to any secondary metabolism or primary metabolism differences within that or between that species? For that, we downloaded the structures of 11 different species from the AlphaFold database. We filtered them for different quality metrics. We used P2Rank, the P2Rank algorithm, to detect binding sites for these proteins. P2Rank is basically predicting the ligandability score for solvent-accessible surface atoms and then cluster points of high ligandability together to arrive at the pocket. And we again filtered that for probability over 0.5, mean PLDT over 70, and mean PAE under 10. And through that, we arrived at about 89,000 different binding sites found in 4 different kingdoms, such as bacteria, fungi, plantae, and animalia. These species that we looked at had quite a differing proteome size, ranging from about 4,000 predicted protein structures to around 48,000. And also their annotated, the number of annotated pockets differs quite a lot. For example, glycine max, which has the largest proteome, as in predicted protein structures found in the AlphaFold database, has one of the lowest number of known pockets. So yeah, these data set of 98,000 different binding sites was then input for binding site comparison. And the aim of this was to characterize the pocketome of different species, cluster the predicted binding sites, and identify the number of unique pockets per species. And for that, we did use 2 different binding site comparison methods. The first of all was PROBIS, which is graph-based. So each of the residues in the binding site is converted into a functional group, and then these graphs are compared via graph matching. And we used the resulting alignment score methods, matrices for as input for the Leiden algorithm. Yes, and through that we arrived at a plot. So we plotted the number of fold-seq clusters to avoid to account for gene duplication within the different species, whereas the number of pocket clusters. And as you can see, this number is dropping from E. coli to different species, plant species such as soybean, maize, and rice. And if you think about the random or the expectation under proportionality, so if every fold would add a new binding site, then this would be a straight line. However, our plot suggests that this, despite there are more in different protein structures, actually we observe a scaling law that's less than proportional, suggesting that there is either like a saturation in different binding sites, or that these new folds are actually invested in different purposes, such as transcription regulation or other purposes. So to kind of put a number on that, we performed the log-log linear fit for the number of Fulci clusters and the number of pockets. And to make sure that this is not an artifact, we also created a null model through degree-preserving randomization. And this, the scaling coefficient for this null model is close to the expectation under proportionality. So yeah, and although this is at least partially driven by the number of singletons, so the number of binding sites within one organism that have no similarity to any other binding site within that species, we can still observe a significant difference between the scaling coefficient for the number of communities versus the number of pocket clusters. And actually, you can also see that the plant species are then more spread, which might suggest that soybean is, this might be due to the bigger secondary metabolism of soybean because it's a leguminose. So we were wondering, what are these singletons, and might this actually be some false positive? And we investigated different physical chemical properties, but also other features of binding sites. And the largest effect size was observed for the pocket probability, suggesting that these are actually either a unique pockets within that species, or that might be false positive pockets that should be discarded. However, we actually wanted to arrive at a global view of the pocketome, so therefore we used a different comparison method, deeply tough, which basically converts each of the pockets to a 3D image with 8 different channels, and then uses a steerable CNN to create a 128-dimensional vector through contrastive learning. So there is binding sites with known similarities and binding sites that are known to be not similar, and through that they are training the embedding space. So first of all, we were interested, how does deeply tough discriminate different ligand classes, so annotated pockets for different ligand tag classes. And as you can see, this is working quite okay for some of the ligand classes, such as nucleobides, nucleoside, nucleotides, also glycan or heme binding sites. But then for other ligand classes, deeply tough does not discriminate them as well. And then we have also this territory where we cannot see as many binding sites that are overlapping with any known binding site. And this needs to be further investigated. So to actually arrive or to actually check what deeply tough discriminates binding sites on, we calculated different physicochemical properties and also other features for each of the binding sites. And as you can see, the binding site discrimination is mainly based on global features such as hydrophobicity and aromaticity, and then other features such as the number of residues does not play as a big role, because this is also expected because this is not an input of deeply tough. And then again, we see this area of binding sites of a lower probability, which might be either false positives or actual allosteric sites. So they are 30% of these pockets are actually the second highest predicted pocket for this protein. So it might be also highly hydrophobic allosteric sites. And at the last, we were wondering, are there any kingdom-specific pockets? So do we have areas of lower entropy? And for that, we calculated the Bind entropy for the t-SNE plot. And we identified one area of lower entropy, which is mainly dominated by kinase domains. So what are the challenges for the binding site comparison? So first of all, there's no formal definition of binding site similarity. Each of the binding site comparison methods comes with different definition of that, like the RMSD, the Euclidean distance, the alignment score, or a simple ligand similarity. Then the definition of binding sites differs. So known versus predicted pocket, we observed that they are often, the predicted pockets is larger than the known pocket. And also within the same fold, we see that there are slightly differing binding site definitions, which is especially a problem for binding site comparison methods that require perfect match, such as syndicate descriptors or also the structural motive search from ISMB. Then, of course, conformational changes are always playing a role, such as the difference between active and inactive states, flexible loops, or also differences in sidechain conformation. And uncharacterized or novel proteins and binding sites are always challenging because they might reside in low quality AlphaFold predictions or out of distribution binding sites for deep learning methods. So with this, I come to my summary. We attempted to generate a global view of the pocketome universe. We discovered a sublinear scaling law between number of pockets and the proteome size. The segregation of pockets based on deeply tough embedding space is based on global characteristics, but fast pocket comparison at a finer detail is still needed. And we identified kingdom-specific pockets that are found in the protein kinase domains. And all of this is accepted for publication in PLOS Computational Biology. And with this, I would like to thank you for your attention and the MPIP Bioinformatics group. And yeah, I'm happy to take questions.
Yeah, thank you. I would like to ask, have you checked the link between the binding site and intrinsic disorder region? Yeah.
No, not yet, but I would assume, because we're filtering for the PLDT and PAE values, so we actually kind of do not have them in the binding sites, in our data set. Yeah.
In one slide, you showed assignment of different pocket type, like for ligand binding, for lipid, for sugar binding. How did you assign those?
You mean this one or this one?
Yeah.
We did use the UniProt as annotations.
So, okay, got it.
I have one question. Each protein, did you take multiple pockets or just one protein? Okay.
No, multiple pockets are allowed per protein.
We have an online question from Ravi Amrol. Do you also include allosteric pockets?
That would be, I think, the same question. So we have, they are not classified for allosteric or catalytic sites, but we allow multiple pockets per protein.
Over here.
Yeah.
Thanks for a really great talk. Did you correlate the variation of similar binding sites to variation in the sequence to maybe pinpoint mutations that are responsible for a specific change in the pocket?
Not yet, but that's a nice idea.
Thank you. Great work. Really nice. I was wondering about the PLDT threshold. You mentioned average. Is that average of the model or average of the pocket?
It's average of the pocket.
Very nice. Okay. And have you considered running F-pocket rescored with Prank?
Yeah.
I think that might be the best.
Yeah, I saw your publication on that. So yeah, that might be the way to go. Yeah, awesome.
Excellent. Great work. Bye.
Thank you for the presentation. What about the cryptic binding sites? Do you have any idea how to deal with this kind of binding pockets, like the ones that are not appearing in the protein?
I mean, there is distinct prediction algorithms for that, and as far as I know, PeterRank does not perform as well for those. But I know that the authors of PeterRank have also different prediction algorithms for cryptic binding sites, but they are not yet included in our work.
You had some trend lines with 4 plants on the right-hand side.
Yeah.
Arabidopsis obviously it's a smaller genome, it behaves differently. Do you think that's a reflection of the annotation quality being much higher there?
Yeah, that might be. So, yeah, that might be. Actually, there's a publication or a bioarchive paper that investigates the difference in AlphaFold structures for different plant species, especially for mice, and they find that they are of low quality. So it might be also that they are, because what we see here, sorry. So yeah, that would be the PLDT and PAE distribution before the filtering. And then you can also see that rice and mice have a bigger distribution than for the others, for example, compared to E. coli. So yeah.
Nice talk. Have you considered all the isoforms in the eukaryotes for this prediction of binding states?
So we used the proteome data from AlphaFold database, which I think does not include all the other frames.
Oh, yes. One more question. Proceed.
Two simple ones. One, now that you showed this, I'm guessing PLDT is average PLDT of the model.
Yeah.
What's the PAE? The average of the whole model or average of the domains?
Average of the pocket as well, but over the matrix, basically. Matrix.
Right, that is average PAE of each pocket.
Yeah.
Very nice. And could you remind me again which features that deeply top include?
It includes hydrophobicity, aromaticity, hydrogen bond donors, hydrogen bond acceptors, and then also presence and absence of an atom. And I think I forgot two. Yeah.
Do you think including bacon data? Yeah, similar question too. There is that prediction. So yeah, that's fine. Great. Thank you.
Thank you so much, Han.